All right, we're on to our last lecture of the first semester. So we should be masters of DNA by now. Deoxyribonucleic acid. It's a nucleic acid because it's found in the nucleus of the cell. And it's deoxyribo because it has deoxyribose as its sugar. Well, now we're going to learn about its buddy, RNA, another nucleic acid. It's got the sugar ribose. That's why it's ribonucleic acid. And we're going to learn about transcription and translation. Those are the two steps of what's called protein synthesis. Synthesis just means to make. So the process of making proteins in the body. So the thing is, DNA has the code for making proteins. Unfortunately, DNA is trapped in the nucleus. So it's got to give its code to its buddy RNA, which RNA can then leave the nucleus, travel to the cytoplasm to a ribosome to make the protein. All right, so we're going to try to get through this fairly quickly. All right, so now some stuff for your notes. RNA, ribonucleic acid. So we're going to go over the differences between DNA over here and RNA over there. All right, so first of all, DNA is double-stranded. It's got two sides. One, two. It's double-stranded, where RNA is single-stranded. You can see it's just a single strand. Doesn't have two strands, just a single strand. DNA has the sugar deoxyribose. You can't really see it in the picture. RNA has the sugar ribose. Here's probably the most important one. DNA has the base thymine or T. Remember, we should know that. Remember, A goes with T and C goes with G. Well, RNA, there's no more thymine. RNA now has the base uracil. So now it's A goes with U. Whoop, went off the screen a little bit there. But C still goes with G. So there's no more T or thymine in RNA. Now it's uracil. A is going to go with U. DNA is stuck in the nucleus, and RNA can travel around the cell. That's why we need RNA. DNA has the code for making proteins that we need to make in our body in order to live. But the proteins aren't made inside the nucleus. They're made outside of the nucleus in the cytoplasm at ribosomes. So that's why DNA has to give that code to RNA. It can then leave the nucleus, head to a ribosome, and make the protein. All right, unfortunately, like DNA, there's only one type of DNA. RNA, there's three types of RNA. Now, if you're like, oh, three is too much to remember, I can only remember two things. Well, okay, if you can only remember two things, the ones you want to focus on are the first one and the second one. The first one is mRNA, which stands for messenger RNA. This is the one that's going to carry the code from DNA, carry the genetic information from DNA in the nucleus, and transport it out into the ribosome for protein synthesis. So this is the guy. It's going to copy the code. It's going to copy what's called a gene in DNA. It's a small segment of DNA. It's going to copy it, and then it's going to leave the nucleus and head to this is the third one, ribosomal RNA. Now remember, if you're going to forget one, this is the one to forget because it's usually the one that's not on the test. Ribosomal RNA, all it is, it makes up a key component of the ribosomes. And remember, ribosomes, you will need to know ribosome. You might not have to know rRNA specifically, but you will have to know ribosome. Ribosomes are where protein synthesis occurs. It's where proteins are made. Now, the middle one here, tRNA or transfer RNA, this one you're going to need to know. I wonder what its job is. Look, it's got a little kind of a greenish bluish ball on top of them, and it's an amino acid. tRNA, its job is brings amino acids to the ribosomes. So messenger RNA, it has the code. It knows how to make the protein. Ribosomal RNA, that's where the protein is going to be made. And tRNA or transferred RNA, it's going to bring the ingredients. It's going to bring the things that you need to make a protein. Because remember, we should know that proteins are made from amino acids. You string a bunch of amino acids together, you have a protein. All right, before I go over protein synthesis, how proteins are synthesized or made, synthesize just means to make. A couple terms here. First of all, there's things called codons. It is a sequence of three, not one, not two, three nucleotides or bases on an mRNA molecule. So here's mRNA right here. This is the guy that copied the code in DNA, so it knows how to make a protein by this code that it's got. And the three bases in a row make up a codon. So right here, GUC, that's a codon. The next one is CAG, that's a codon. You can see right here, GAG, that's a codon. CCA codon and UAG, those are codons. So there's 5 codons here. It's 1, 2, 3. Now, also the tRNA, which you can see right here, remember, it's the guy that carries amino acids, and it's carrying the specific amino acid, glutamic acid, and that's one of the 20 amino acids. You put them together, you make proteins. It has what's called anticodons, or some people call them anticodons. It's 3 bases again, and his one is CUC for this specific tRNA. And they're going to match up codons and anticodons are going to match up according to the new base pairing rules. So G is going to go with C. U goes with A, because remember, there's no more T in RNA. C still goes with G. C is going to go with G. A goes with U. G goes with C. And you can see this one. C goes with G. It always does. U goes with A. C goes with G. So this code, these codons, are what tells the tRNA which amino acid to drop off. So every time there's a glutamic acid on top, it's always going to have the anticodon of CUC on the bottom. So codons, sequence of 3 bases or 3 nucleotides on an mRNA. Anticodons, a sequence of 3 bases or 3 nucleotides on the bottom of a tRNA. All right, amino acids and their role. This is very simple. Amino acids are the building blocks of proteins. So proteins are made from amino acids. There's 20 different amino acids. You will not need to know them. Here's some of them over here, or all of them. Phenylalanine, tyrosine, arginine, lysine, aspartic acid. Anyways, so remember, it's the order of the bases in DNA, the A, T, C's, and G's, that's going to determine the order of the codons in the mRNA. And it's the order of those codons. Remember, a codon is a 3-base sequence, like CAG, that's a codon. AAU, that's a codon. It's the order of those codons that's going to determine the order of the amino acids that's being added to your protein. And it's the order of those amino acids in your protein that determines the function of that protein. So if you have brown hair, that's because your proteins, your amino acids, are in a certain sequence. If you have blue eyes, it's because the amino acids are in a certain sequence in your proteins, and it's giving you those traits. So that's the function of amino acids. They are what proteins are made out of. All right, the nucleus, where it all begins with transcription. The nucleus contains the cell's DNA. So that's where DNA is found. It's stuck in the nucleus. So here's DNA right here. It's stuck within the nucleus of the cell. Transcription is the process, and I'm going to go over it in the next slide. It's where RNA is copying the code in DNA and forming a messenger or mRNA. Now, the reason why we need the mRNA, because mRNA can leave the nucleus, head to the ribosome over here. Here's the ribosome. Now it's got the code from DNA, so it knows the sequence of A, T, C's, and G's. It got them in codons, 3 bases on the mRNA, codons. So it's got all that. It takes it to the ribosome, and now a protein can be assembled. So transcription occurs in the nucleus. It makes a messenger RNA. That messenger RNA can now leave the nucleus. Remember, DNA cannot. DNA is stuck in the nucleus. So this is transcription, the first step in protein synthesis. I'm going to go over it more on the next slide. All right, so this is the first step of protein synthesis, how proteins are made. It's called transcription. It occurs in the nucleus because that's where DNA is found. All right, so you're not going to need to know, just know the basics. So first, remember, DNA is double-stranded. In order to make the messenger RNA, for RNA to copy the code, it needs to unzip, kind of like on DNA replication. So here's your messenger RNA right here being constructed. In blue, that's the DNA. So DNA unzips and exposes the bases, the A, T, C's, and G's. And then RNA polymerase, you're not going to need to know that. It's an enzyme. It's going to attach free-floating nucleotides to their appropriate base. So when it sees a T, it's going to put an A. When it sees an A, it's not going to put a T because there's no more T in RNA. It's going to put a U. C, it's going to put a G because G always goes with C. G, it'll put a C. A, it's going to put a U, uracil. There's no more T in RNA. G will put a C. A will put a U because there's no more T in RNA. A will put a U. T will put an A because there's still A in RNA. And boom. So this is going to attach right here. This is going to attach right there. And then this entire mRNA, it just got built in the nucleus by copying the code in the DNA. T goes to A goes with U, C goes with G. And this entire strand right here, the mRNA, is going to leave the nucleus and head to the cytoplasm because now it has the code. It just copied the code from DNA. And then once it's done, DNA will go right back together, being double stranded again, being a double helix. All right, so we just did transcription in the nucleus. That's where we made this messenger RNA right here. We made this messenger RNA during transcription in the nucleus, where we copied the code in DNA according to the base pairing rules. So A went with U, C went with G, C went with G, T went with A, and so on. So we made this messenger RNA in transcription. Now we're actually going to make the protein. Here are the amino acids. They're bonded together. That's your protein right there. Here's your tRNAs. Remember, their job is that they carry the amino acids to the ribosome, which is right here. All right. So the ribosome is going to read the codons U, G, G, and a tRNA that has the corresponding anticodon is going to match up with it. So U goes with A, G goes with C, G goes with C, and then it dropped off its amino acid. Here's this codon. A is going to match up with the anticodon of U, U, U. Because remember, there's no more T in RNA. A goes with U. And it's going to drop off its amino acid, which happens to be lysine. The next codon is GAU. According to the base pairing rules, G goes with C, always goes with C. A goes with U now because there's no more T in RNA, and U goes with A, and it's going to release its amino acid asparagine. And the last codon right here is U, U, U. So the tRNA that has the matching anticodon, U goes with A, U goes with A, C goes with G. And it's going to release drop off its amino acid, phenylalanine. So this is the process. tRNAs dropping off their amino acid and building this protein right here. So this is translation, reading the code from the mRNA at the ribosome, and tRNA matching its anticodon, matching up with the codon, and dropping off the specific amino acid in the specific sequence that it needs to be in. So there's translation that occurs at the ribosome. All right, I think this is the last slide because I'm up over 12 minutes and I don't like to go that long. But anyways, how to read a codon chart. Here's our codon chart right here. Here's our messenger RNA here at the bottom. I'm gonna show all the codons. Remember, codons are three bases. So there's our first one. There's our second one. There's our third one, AAG. There's the fourth one, AGC, and there's the fifth one, CUU. All right, so first thing I'm going to do is, here's our tRNA. What's the anticodon that would go with G? UA. Well, G goes with C, U goes with A, and A doesn't go with T because there's no more T. A goes with U. But the question is, what amino acid is this? To do that, we got to look at the codon chart. G, U, A. So I go over here. You always start in the very middle, right here. And then the first base is a G. So I go this way to the G. Now I know it's in this region right here. So I know it's going to be in here somewhere. The next one's a U. So I go from the G to the U. Boom. And the last base is an A. So I go from here to the A. Valine. So I know that amino acid right there is valine. Let me try another one. Now I'm going to let you try a couple. All right. So the next one is, the codon is UGC. The anticodon would be A. U goes with A. G always goes with C. And C goes with G, always. All right, so now we're going to look at the codon. We don't look at the anti because it's a codon chart, not an anticodon chart. UGC. I'm going to start right in the middle. Boom. All right, the first base is a U. So I'm going to go this way to the U. Now I know it's in this quadrant right here. The next base is a G. So then I go from the U to the G right here. And the last base in this codon is a C. So I go from the G to a C. Cysteine. This amino acid right there is cysteine. All right, so now I want you to try. I'll do one more. AAG. Let me erase this. AAG codon. I got no more tRNAs, but the tRNA anticodon would be UUC. But anyways, what amino acid would show up with AAG? I'm gonna start in the middle. Boom, right there. The first base is an A, so I go from the middle to an A. So now I'm staying in this quadrant. I'm not gonna end up going over here. I'm gonna stay in this little fourth right there. The next one is an A, so I go from the A to another A. So I'm not going to A over here or A over here. I'm gonna continue to go this way. And the last base is a G. So I go from the A to a G. The next amino acid would be lysine. All right, so I want you to try the next one. A, G, C. Let's see what you come up with. All right, let's see how you did. So the next codon was A, G, C. So once again, we're going to start in the middle. The first base is an A, so I'm going to go from the middle to the A. The next base is a G, so I'm going to go from this A to this G. I'm not going to this G or this G or this G up here. You stay in this little quadrant. And the last base is a C, so I go from the G to the C. And the next one is serine. All right, I'm going to give you one more shot. C, U, U. Let's see how you can do. All right, the last codon is C, U, U. Remember, the anticodon would be G, A, A. That would be on the bottom of the tRNA. But CUU, what amino acid will show up with CUU? First base is a C. I start in the middle. I go to the C. Boom. So now I'm going this way. I'm going down and to the right. Next base is a U. I'm going down and to the right. Staying in this little quadrant. There's a U. The last base is another U. And U. And we got leucine amino acid. So hopefully you got that. All right. We got one more slide and we're done. All right, this is just a review. I'm not going to read all this because I've already gone almost 17 minutes. That's way longer than I like to go. But remember, in college, 17 minutes, just getting started, bro. You got another 45 minutes or so of lecture. So feel free to pause this video, take down some notes because quiz time is coming right now.